Students, welcome to BioWise with Aanchal. Now today we are going to study about a chapter that is chapter number 5 of standard 9 biology, that is pollination and fertilization. Now you must have studied about pollination and fertilization in your 8th standard when we were studying about the flower chapter, remember? Even if you don't remember, it's okay. But before proceeding with the chapter, like, share and subscribe my channel for more updates. Now in this chapter, we are going to study about pollination, types of pollination, self-pollination, cross-pollination. Nature favors cross-pollination. This statement is very important. Agents of cross-pollination. So this is all we are going to study. Now before coming into picture, just pause the video here. Write down the definition of pollination, what you know. What is pollination? I could see some people, some messages, very few, but still I can see some messages. Thank you so much for writing in the comment section. Now, what is pollination? Pollination is a process of transfer of pollen grains from anther of a flower to the stigma of the same flower, या फिर different flower of the same plant, या फिर different flower of the different plant of the same species. The pollination will be successful only when the pollination takes place between the flower of the same family, or you can say same species. Okay. Now you can see here, pollination is what? Transfer of pollen grains from the anther to the stigma. Check how many of you have given the right answer in the comment section. Now, types of pollination, when you talk about, it is self-pollination and cross-pollination. Self-pollination means the plant, same flower, that is, one flower is there only the transfer is happening. That is called self-pollination. That flower can belong to the same plant, right? Two flowers on the same plant can also go for self-pollination. So there are different terms that we use, like autogamy. Autogamy is the self-pollination where the transfer of pollen grains from the anther of a flower to the stigma of the same flower is called autogamy. Geitonogamy is considered to be self-pollination only, but in this case, there are two separate flowers on the same plant. So transfer of anther, that is transfer of pollen grains from the anther of one flower to the stigma of the other flower, but on the same plant. That is called geitonogamy. Okay. Allogamy or xenogamy is the transfer of pollen grains from the anther of a flower to the stigma of the other flower on the different plant of the same species. Okay. If you need to do some hybridization, that is, you can take the same family plants, maybe distant, but you can have the hybridization. Okay. We will talk about, like, for example, autogamy, maybe you have direct autogamy and indirect autogamy. Direct autogamy means the transfer takes place automatically, that is without the involvement of any pollinators. But indirect autonomy may you need insects like bees and all to pollinate the flower. I hope the concept is clear. Let's move to the next one. That is, what is the advantage of self-pollination? Now, self-pollination, first of all, the parental characters are preserved indefinitely. If you get a very good variety, you can go on multiplying it again and again and again. So that is your self-pollination. Small quantity of pollen is sufficient. Okay. Flowers need not to be very large or showy because you're not attracting any pollinators. You don't need a large or showy flowers. And there is no need of scent or nectar. So plant is actually saving lots of energy in not making a big flower, a scented flower, or a flower which has nectars to attract insects. Great economy of the part of the plant as lots of nutrients are used in the production of pollen scent, nectars, and large and showy colorful petals. So it's economical for the plant. Then what are the disadvantages? Now, when you talk about disadvantages, the variety, because of being generation after generation, the same variety is being transferred, there is a weakening of the variety of the species. In some plants, the vigor of the plant is also lost. That is, the ability to produce seeds or crops is also reduced. Weaker or defective characters or recessive genes cannot be eliminated. You know that recessive genes are expressed if uh, if there is a cross between a very close plant, or you can say same family or same species, same parental uh, genetics if it is there. So in that case, the recessive genes are highlighted. Okay, now I'll give you a very good example. You've heard about hemophilia. Hemophilia is a genetic disorder, and generally it is seen in the royal family because the marriages are happening within the family itself. The royal blood is flowing in the family, so the genes are also flowing in the same family. So the recessive genes are highlighted. So the diseases are highlighted. In case of consanguineous marriages, also, that is marriages between the cousins, there also we see that the chances of recessive genes are highlighted more. Okay. There is no new varieties formed. Since it is a repetition of the same variety, there is no new variety formed. Advantages of cross pollination is healthier offsprings are produced, seed produced are many, and new varieties are produced. You get hybrids, better yield, better varieties, right? Now, disadvantages of cross pollination. It is not certain. You don't know whether the pollination will happen or not happen. There will be wastage of pollen grains. Now, suppose there is a wind and there are lots of pollen grains in the wind. You don't know that out of those many million of pollen grains, how many are actually going to germinate, or how many of them are actually going to get transferred to the stigma. After getting transferred to the stigma, also, how many compatibility is there, how many matches are there, then how many pollen tubes actually is germinated from the pollen. So that is not sure. So it's totally uncertain. It's highly uneconomical for the plant because the plant has to produce lots of varieties of material like large, showy petals, nectars, scent to attract the insects. Actually, nature favors cross pollination because cross pollination helps in a better offspring, a recombinant offspring, or a better offspring. So nature favors cross pollination. And how we are going to prove this statement, we are going to see in the next slide. Now here, if you see, like why nature favors cross pollination, so there are certain terms like unisexuality. You see that there are plants called like papaya, cucumber. There we have a plant of one gender, like monoecious, dioecious. We have studied in the previous chapter, flower chapter. What is monoecious plant? Write in the comment section. And what is dioecious plant? Write in the comment section. Okay. Papaya and cucumber are monoecious or dioecious. That also write in the comment section. Now, next term is dichogamy. Dichogamy means that is either of gynoecium or androecium is going to mature first. And then, obviously, if the other better half is not matured enough, then self-pollination will not happen. Cross-pollination is the only option left, right? Like protoandry, you see that in sunflower, sweet pea, okra, that is bhindi. You see that the androecium matures first. That is called protoandry. Similarly, in custard apple and people plant, you'll find that gynoecium matures first. That is protogyny. Cell sterility. In certain flowers like sunflower, the ray florets, the yellow color ka ray florets, they are cell sterile. They are sterile flowers. They are neuter flowers. They don't have the male and the female gamete present on it. So they are self sterile. And the uh, disc floret is basically the fertile flower present there. Orchids also, they are self sterile. That is, they cannot pollinate, or they cannot make the pollen of the same plant germinate on it. That is called self sterility. Hercogamy, there is a mechanical barrier between androecium and gynoecium. So the anther do not touch the gynoecium. So then in that case, the pollen from that anther can never be transferred to the stigma of the same flower. So self-pollination is ruled out. Heterostyly, in primrose and oxalis, you'll find that the length of the androecium and gynoecium is so varied, so different, that they are not able to fertilize each other, or they are not able to pollinate each other. I should not use the term fertilize, but they are not able to pollinate each other. So in that case, again, self-pollination is ruled out. Again, with examples, if you see, now see, unisexuality, if you talk about male and the female, 1 minute. Yes. Now here you can see the male and the female flower are born on separate plants. So papaya has got a male flower, a female flower, but on the separate trees. Similarly, cucumber also has got a male flower and a female flower on a separate plant. Right. Protoandry in sunflower anthers mature first. Protogyny means stigma matures first. Self-sterility pollen of the flower cannot fertilize the stigma of the flower, like ray florets of the stigma of the sunflower, orchids. Then hercogamy is a mechanical barrier. You can see this is a pansy flower. So there is a physical barrier, a petal, an extra petal is there in between, which does not allow the pollination, self-pollination to occur. Heterostyly means the length of the androecium and the gynoecium is too varied, so self-pollination does not happen. Please remember, unisexuality is seen in papaya and cucumber. Dichogamy, protoandry, and that is seen in sunflower, sweet pea, okra. Protogyny example is custard apple and people. Self-sterility is seen in the ray florets of sunflower and orchids. Hercogamy is seen in pansy flower and iris. And heterostyly is seen in primrose and oxalis. Okay, so this is what actually these conditions, if you see, they are actually not allowing the what to say, the process of self-pollination to occur. Understood? So if self-pollination is not possible, then obviously cross-pollination will happen. Correct? Moving to the next, agents of pollination. Now, if you see about the agents, there are two variety of agents we have. One is a living agent and one is a non-living agent. So living agents are called the biotic factors. So bird, insect, bats, elephant, they are all are the biotic agents which help in pollinations. So they are called pollinators. And abiotic factors may wind and water. That is a very good example of abiotic factor. So there are specific terms that we use for wind pollination, water pollination, bird pollination, insect pollination, and so on. Right? So first is we are talking about the insect pollinating flowers. You can see for a flower to be pollinated by insect, if it is a daytime flower, it has to be very bright and colorful. It has to produce nectaries so that the insect gets some food to eat. Others, why will that insect come to you? Right. Another thing is that if it is a night flower, then also it should be bright white or it should have very beautiful scent. And that is the reason most of the white flowers that blooms at night has a fantastic aroma, isn't it? Right? So they are basically large, they are brightly colored, they are scented, they have nectars, they have sticky pollen and stigma, so that the insect, if brings the pollen or if the insect sits on the anther or on the flower, the pollen can stick to the uh, that is on the insect body. And stigma is also sticky so that any pollen from the insect can also stick to the stigma. And they grow in clusters so that it attracts the insects. So this is all the characteristics of entomophilous flower. So hibiscus is a very good example of entomophilous flower. Right. Now children, remember the characteristic features. You can use your common sense also to write about the characteristic features. Next is anemophilous. Anemophilous is basically wind, pollinated by wind. So flowers are small, not that brightly colored because you don't need any pollinator to get attracted towards it. The stamens are long, feathery, and they hang out, and large and loosely attached anthers are there. Large quantities of pollen grains are produced, and they are light, dry, and smooth pollen grains which are produced so that they can be easily carried by the wind. Okay, so these are the characteristics of the wind pollinated flower. Coming to the next is the hydrophilous flower. Now, hydrophilous flower, the best example is the Vallisneria. Now, Vallisneria is a plant that grows under the water. So, in this case, what happens? There are male, you can see this long twig, this long twig. This is basically the female flower. Okay, and this is the male flower. So the female flower, they are actually, what to say, they're long enough to reach the surface of the water, and the male flower produces the, what to say, the pollens which come out of this, and even the male flowers leave the plant and they go and float on the surface of the water, so that when they meet the female flowers, they transfer the pollen grains to the stigma of the female flower. Once the female flower gets the pollen, develops into the fruit, again this twig just comes under the water and the fruit develops under the water. This is a very good example of the pollen, that is water pollinated plant, Vallisneria. Understood? Next is artificial pollination. Artificial pollination, basically, if you study in your 10th standard genetics, this is you're going to study next year. So genetics mein what Mendel did, that is what artificial pollination is all about. In this, there are few words like emasculation. Emasculation is a process of removal of anther from a flower, from a bisexual flower, so that there is no anther, no pollen, no self-pollination. And whatever anther you want, that can be dusted over the stigma of the flower. So the artificial pollination, here human becomes the pollinator. Understood? Human becomes a pollinator here. Now, Gregor Johann Mendel, the father of genetics, he worked on pea plant. Pea is a closed flower, clistogamous. Okay? So, closed flower mein chances of cross-pollination is not there. So, in order to have a cross-pollination according to his choice, that which trait he wanted to study, he did artificial pollination. Understood? Any doubt? Put it in the comment section. Coming to a very important part of the chapter, that is called fertilization. Now, children, you all know what fertilization is. We are studying fertilization, I think, from 6th standard. Fusion of male and the female gamete. Fusion of the male and the female gamete to produce a zygote is called fertilization. So, union or fusion of the male and the female gamete to produce a single cell zygote, which later develops into the entire embryo. In case of human, the entire baby. In case of the plant, the seed. That is all that is called fertilization. Now, when we talk about the fertilization process, understand this, okay? This is really important. You can see here, right? The pollen grain here, once the compatibility match is tested, matlab, the pollen belongs to the same species of which the stigma is, then the pollen tube germinates. Once the pollen tube germinates, it has two nucleus. We don't use the word sperm, right? Male gametes, that will be better. So here, uh, it's a better option. You can use sperm cells, use sperm nuclei, it's given most of the textbooks. But still, if you use male gamete, it will be a little bit more precise, right? So now what happens, there is a tube nucleus and there is a generative nucleus. We'll study about the pollen structure in the next slide. This pollen tube germinates and transfers these two male gametes into the female ovule, right? That is present inside the ovary of the flower. Now what happens? These male gametes fuses in two places. First, it fuses with the egg cell and then it fuses with the secondary polar nuclei. The secondary polar nuclei is again formed because of the fusion of two polar nuclei. So now two polar nuclei fusing to become a secondary nuclei. Secondary nuclei fusing with the male gamete to form the endospermic nuclei. There are fusion of three nucleus. So the process is called triple fusion. And since there are two male gametes, two fertilization happening, it is called double fertilization. Understood what is double fertilization? We'll do it in detail. Now look here. First, understand the structure of the pollen grain. Now, the pollen grain basically consists of a spiky outer covering, which is called the exine, and intine, that is the inner layer of the pollen grain with cytoplasm and two types of nucleus. One is called the tube nucleus, which helps the pollen tube to grow towards the ovary. And one is generative nucleus, which actually contains the male gametes. Understood? Now, this tube nucleus is going to direct the pollen tube to the ovary. Behind, the generative nucleus will go on moving and dividing into two male cells or male gametes, which will later go and cause double fertilization. Okay, this is the microscopic view of the pollen grain. You can see here, right? Coming here, this is a huge written content. You can take a screenshot if you want. Okay. Now here, if you see, pollen grain is a male gametophyte. Okay. Pollen grain is a male gametophyte of the flowering plant. It is produced in the anther and is transferred to the stigma during pollination process. Okay. Now, when you talk about the components, the exine, the outer layer of the pollen grain, that is thick and tough, made up of sporopollenin. It's a component present in the outer layer of the pollen that is called sporopollenin and has sculpturing or ornamentation like spikes present on the surface, which helps it to stick to the stigma. Have many pores called the germ pores that is present in the exine. Now, intine is basically thin, smooth, and continuous, made up of cellulose and pectin, lies beneath the exine. The exine may you have sporopollenin, and in the intine you have cellulose and pectin. Then the vegetative cell or the tube nucleus. The tube nucleus is generally for the growth of the pollen tube towards the ovary. It is large cell with lots of cytoplasm, and it contains a very large nucleus, helps in the growth of pollen tube. Generative nuclei generally is a smaller than the vegetative cell or the tube cell, and it is having a dense cytoplasm, divides to form two male gametes. So out of the two, tube nucleus and the generative nucleus, which one forms the male gamete? It is the generative nucleus which forms the male gamete. Okay. Now, tube nucleus is the nucleus of the vegetative cell. Generative nucleus is the nucleus of the generative cell. Is this clear? So, two male gametes are basically formed from the division of the generative cell. They fuse with the female gamete and the polar nuclei during fertilization. So this is what the structure or components of pollen all about. You can see the structure here. You have exine, you have intine, you have the tube cell containing the tube nucleus, you have the generative cell containing the generative nucleus, and finally the pollen germination takes place once this movement, that is the growth of pollen tube, is a chemotropic movement. Now again a big word has come. This word is there in your 10th standard in the chapter Chemical Coordination in Plants. Chemotropic means the stimulus of growth is chemical, and the when we say tropic movement, it is directional growth. So if the pollen tube is growing towards the ovary, there is some chemical in the ovary which is attracting this growth. So here stimulus is chemical and the growth is towards the ovary. This is called a chemotropic movement. If you remember, fine. If you don't remember, you have to learn in 10th standard again. Okay, so this is all the components of the pollen. Now, when you talk about the ovule, now ovule, this ovule I have made in this way, right? The ovule can be made this way also. It can be made this way also. So whatever be the size and shape. Now, if you see this way, wherever is the opening is the micropyle. Wherever is the opening is the micropyle. Through the micropyle is also there in the seeds. We'll study again. This is the same micropyle which will develop as a small pore in the seeds from which the water enters during germination. So now there is a small embryo sac. This is the embryo sac. In the embryo sac, there are total 3 plus 3 plus 2. That is 3 plus 3 plus 2 is what? 3 synergids plus 2 polar nuclei plus 2 I'm so sorry, 3 antipodal cells, 2 polar nuclei, 2 synergids, and 1 the queen, that is the egg cell. So total you have got 8 cells present in the embryo sac. Okay. Now this is how, and then we talk about the fertilization in the next slides. Hilum is the point of attachment. Here you can see funiculus is a stalk of the ovule attached to the placenta. I hope you remember the flower chapter. Then you have the secondary nuclei. This is after the fusion of the polar nuclei. Now always remember all these 8 cells are haploid. What is haploid? That is containing half set of chromosomes. Okay, so that upon fusion, they will have diploid or the double set of chromosomes. In basic, what to say, alphabets, we can denote this as N. These are all haploid. Diploid is denoted as 2N. Okay, the haploid is N, diploid is 2N. All these female gametophytes are all haploid. Yes. Now, when you talk about double fertilization, see, this is the handmade drawing for the process of fertilization. You can see here the pollen tube, the pollen grain germinates, and the male gamete, and you have the tube nuclei moving here. This male gametes divide into two and forms two male gametes. So here they have given an extra, it should be only three. Okay, these are all antipodal cells. What are they? They are antipodal cells. Okay, antipodal. You can see here, these are all antipodal cells. Okay, then you have the synergids. Synergids are the two sister cells present near the egg cell. Okay, they are the two sister cells present near the egg cells. These two are the synergids. The central one is the egg cell. This one is the egg cell. Okay, this is so after fusion of the egg cell and the what to say egg cell and the male gamete, you have the formation of zygote. So we'll not write zygote over here. Okay, we'll write it later. So this is this is helping in the formation of zygote. Clear? So what is double fertilization? Double fertilization, you can say that there are two fertilization happening. One is what? The male gamete with the egg cell to form zygote. And the second one is the male gamete with the secondary nuclei to form a triploid 3N condition, triploid endosperm. Okay, so there are two fertilization happening, so double fertilization. Triple fusion means the two nuclei of the polar nuclei, the polar nuclei will fuse to form a secondary nuclei. That will fuse with the male gamete to form an endospermic nuclei. So total there are three nuclei which is actually getting fused. So it is called triple fusion. Understood? Put it in the comment section, children. I hope you have subscribed the channel. You have reached till this place. That means you're understanding what is going on in the class. So it's better you subscribe it. Yes. Now, double fertilization versus triple fusion. Double fertilization is the fertilization process in angiosperms, and triple fusion is the fertilization of the two polar nuclei in the embryo with the single male gamete. Okay, single male gamete. Fusion of the embryo sac with the two male gametes, and here it is one of the two fertilization. So triple fusion basically is a subset of double fertilization. It forms a diploid zygote and it forms the triploid endosperm. Okay, it forms a triploid endosperm. Produces a diploid zygote as well as a triploid endosperm. In this case, it is only triploid endosperm. Okay, now what is the fate of the floral parts? So if you have to talk about the fate of the floral parts, you can see here that the sepals, petals, stamen, and style and stigma, they generally wither away. They fall off. Okay. Ovary becomes the fruit. Ovule becomes the seed. The egg, the zygote, that becomes the zygote. Funicle becomes the stalk of the seed. Micropyle, that is also the micropyle of the seed. You can see a small opening here, which helps in the formation, is absorption of water and diffusion of gases. Then nucellus, basically the perisperm. Outer integument testa, that is outer coat of the seed, that is outer layer of the seed coat. Tegmen is the inner seed coat. Then synergids, they degenerate. Secondary nucleus forms endosperm. Endosperm is not present in all the seeds. It is present only in the endospermic seeds and certain monocots like maize. Maize where you have endosperm. Antipodal cells and all, they degenerate. Okay. Fate of the ovary. Ovary wall becomes the pericarp, ovule seed, funicle stalk, hilum remains hilum, nucellus perisperm, micropyle, outer integument testa, inner integument testa. Please remember this. It will be easy for you. So what we studied about pollination, what is self-pollination, what is cross-pollination, what are the agents of pollination, what is fertilization, right? What is the step of fertilization? What is double fertilization? What is triple fusion? Right? So this is all that is important for this chapter. So time for practice. Draw a mind map for the lesson. It will help you to analyze whatever we have studied today. Differentiate between self and cross-pollination. Differentiate between the wind pollinated and insect pollinated flower. Write down in the comment section the scientific term used for wind pollinated flower and insect pollinated flower. Write down fast in the comment section. Let me see how many comments I get. And draw a neat label diagram of germination of pollen grain on the stigma followed by fertilization. I have already given you the diagram. You can draw it easily. That's all, children. Thank you so much for being a patient listener, and write in the comment section whether you understand what I teach or not. Okay? For any doubt, if you want any video on any topic, put it in the comment section. I will do the needful. Happy learning, children, and stay connected with BioWise with Aanchal. And do not forget to subscribe the channel. Chalo, happy learning.